Enteroids are three-dimensional organoids derived from intestinal epithelial cells. They are ideal for the study of complex physiologic interactions, cell signaling, and host pathogen defense. Our protocol describes how to culture human enteroids after isolating intestinal stem cells from patients undergoing bowel resection. Co-administration of lipopolysaccharide in our culture media causes an inflammatory response in the enteroids, leading to histologic, genetic, and protein expression alterations similar to those seen in human necrotizing enterocolitis. Given that the study of necrotizing enterocolitis and potential therapeutic agents is ethically challenging in human subjects, especially children, it is highly desirable to utilize this novel, biologically relevant enteroid model of necrotizing enterocolitis using human neonatal tissue. The main advantage of this technique is that enteroids are able to recapitulate the multiple cell types of human intestinal epithelium and are able to overcome the recognized limitations of animal models and cell-based systems. Helping to demonstrate the procedure will be Christie Buonpane, a surgical resident, and Carrie Yuan, a laboratory technician from our laboratory.
At the time of collection in the operative suite, place the human small intestinal tissue sample in cold DPBS. Wash the specimen in the cold DPBS until it is clear of blood and stool. Store the specimen in RPMI 1640 medium at 4 degrees Celsius until ready for crypt isolation. When ready to proceed, check again to ensure that the specimen is clear of stool and blood. Using delicate dissecting scissors, remove any excess fat.